Hello everyone and welcome back for more human physiology. This time we are going to take some concepts that we established in the previous video and we are going to specifically apply it to the movement of water across the plasma membrane. Of course, we are talking about osmosis. So without further ado, let's go ahead and see what we got here. Now, the most important thing that we learned in the last video that we're going to apply to water is that for passive transport, molecules like to move down their concentration gradient. They move from areas of high concentration to areas of low concentration. Water is not going to be any different here. Water wants to move from areas of high water concentration to areas of low water concentration. But here's the sticking point. This isn't usually a problem for us if we're talking about solutes like sodium or potassium or glucose, because if we have a given solution, we are likely going to know those concentrations. Those concentrations are going to be given to us. Water concentration is not something that is typically kept track of. It's not something that you're just going to be given. It's the solvent. We know it's the majority component in solution, so it's usually a lot more efficient to just keep track of all the solute concentrations and then just leave the water concentration as is. So we aren't usually going to know the water concentration, and that's going to make determining whether osmosis occurs or not and in which direction osmosis occurs a little bit more complicated. So anytime you're dealing with our bodily fluids, ICF, ECF, or IV fluids, which we'll talk about later, we're really just talking about keeping track of solute concentrations and not solvent concentrations. So that doesn't mean we can't do it, though. What we're going to try to learn here is we want to be able to see a connection between total solute concentration, not just one or two individual solutes, but all the solutes that are in a particular solution. We want to see the connection between all those solutes and their total concentration and the water concentration. The idea here is that the more stuff being solutes, the more stuff that is dissolved in water, the lower the water concentration will become. And the less stuff that's present, the higher the water concentration is going to be. And if this all sounds familiar, you'll recall that we actually looked at this relationship a little bit back in Chapter 2. So the higher the solute concentration, the lower the water concentration, and vice versa. So this is a diagram from your textbook that illustrates what osmosis is all about. So before we can really determine what's going on with osmosis, we have to establish a couple of rules and boundaries. First, let's look at the picture. We have a before and an after picture. So for now, let's go ahead and ignore the after picture and let's just pay attention to the before picture. Now, this picture is not my favorite, but I can kind of explain what's going on here. We have a beaker that is essentially split up into two different chambers of equal volume. What is doing the splitting here, this red dotted line, is meant to be a semi-permeable membrane. So it's not going to behave exactly like the plasma membrane, but we can say that it's good enough. So in the two different chambers, we have two very simple solutions. Both solutions utilize the exact same solute. It's not specified here what the solute is, but let's just go ahead and say that it's glucose. So if you look at the two chambers here, we'll say chamber A on the left and chamber B on the right. Clearly, chamber A has a glucose solution that is much less concentrated than the glucose solution in chamber B. You can just use your eyes and see that that must be the case. Each chamber has an equal volume of solution, but chamber A has fewer individual solute particles compared to chamber B. Now, ordinarily, what would happen here, if we're talking about the glucose, is that the glucose molecules would undergo either simple diffusion or facilitated diffusion. If we're talking about glucose, it would be facilitated diffusion. And the glucose molecules would move from the right side to the left side to establish equilibrium. There is a concentration gradient here for glucose that compels glucose to move from the right to the left. Now, if glucose is able to do that, there should be no reason for osmosis to occur. There's no need for water to move if the solute itself can move and reestablish equilibrium on both sides. But here's when osmosis does occur. Osmosis will occur here because glucose, we are going to assume, cannot move across the membrane. So even though we have a concentration gradient for glucose, glucose wants to move from chamber B back to chamber A, it cannot do so. It cannot cross the plasma membrane. So we've got this concentration gradient that cannot be alleviated just by the movement of glucose. So osmosis is going to occur in cases in which the solute cannot move across the membrane, but water can. So we're going to assume that this semi-permeable membrane is not permeable to glucose, but is permeable to water. So we've got a concentration gradient for glucose on either side of the chamber, but we also have a concentration gradient for water. Water is at a higher concentration in chamber A compared to a lower concentration in chamber B. Remember, the more stuff that is in solution, the less the water concentration is going to be. So glucose wants to move from right to left, while water wants to move from left to right. So if we go now to the after picture, after water has had a chance to move from the left to the right, you'll notice that the volume in chamber B has gone up because water has moved into that chamber, and the volume in chamber A has gone down because water has moved away from that chamber. But look at what has happened to the concentrations of glucose on either side. Just based on, since we don't have a number to work with here, just based on how far apart or how spaced apart these glucose molecules are, we can get a sense that we have approximately equalized the concentration of glucose on either side of the chamber. You'll notice that both chambers have the same number of glucose molecules that they started with, but the concentrations are now equal because with osmosis occurring, we have changed the volumes, which has also changed the concentrations. Remember, concentration is not just about the number of particles, it's also about the volume of that particular solution. We have increased the concentration of the solution in chamber A because we have decreased the volume, and we have decreased the concentration of chamber B because we have increased the volume. So the thing to remember here is that osmosis will only occur if you've got a solute concentration gradient that cannot be alleviated by either simple diffusion or facilitated diffusion. Usually we're going to be talking about facilitated diffusion there. So after this is all done, the end result is that we change the water volume and therefore concentration without a change in the number of solute particles on either side of the membrane occurring. Okay, so now that we've seen a specific example of osmosis occurring in more of an in vitro or outside a physiological boundary occurring, let's get a couple of notes out of the way. So we know that in the case of cells, water has to move through aquaporin channels. And you may remember before that I said that channels may be open or closed. So in the case of aquaporin channels, you don't really have to worry about that because these channels we can assume are always open. Basically, if a cell has aquaporin channels, it's going to be able to participate in osmosis. Most cells in the body do have aquaporin channels, so it's not usually going to be a case of you having to worry whether this cell has aquaporin channels or this cell does not. So in most cases, you can assume that every cell is going to have an aquaporin channel. It's not going to be until we start looking at kidney physiology before we really start to look at examples of, okay, these cells do not have aquaporin channels, and for that reason, they cannot participate in osmosis. So ultimately, what we are going to be concerned about with osmosis is water moving across the plasma membranes of cells. Now, the way we've discussed it, we make it sound like osmosis is something that needs to occur all the time, but that's actually not the case. Since water does move from high water concentration to low water concentration, What that's going to mean is that if osmosis does occur, we are going to change the water concentration and therefore the fluid volume in either the ICF or the ECF, depending on the direction in which water is moving. So we really don't want that to occur. You're going to see in the next few slides what will happen to the shape and the volume of a cell if osmosis does occur in great quantities. So we don't want too much water to leave the cell, and we don't want too much water to enter the cell. Another way of putting this is that if we want to make sure that we are maintaining the correct amount of water both inside the cell and outside the cell, what we really want is the concentration of water inside and outside the cell to be equal at all times. In other words, we want to establish osmotic equilibrium. That is the name of the game. Okay, so what osmosis is all about, when you're trying to determine when and how osmosis occurs, you're going to have to keep track of all of the appropriate and relevant solutes in a particular bodily fluid. Now, this is, like I said, this isn't just going to be one solute. It's not going to be just sodium or just glucose. In the previous example we looked at, that was the simplest example we could have looked at because it was just one solute. It was just glucose. We know that bodily fluids are complex solutions. They have many, many, many different kinds of solutes simultaneously dissolved in water. Sodium, potassium, chloride, calcium, bicarbonate, proteins, glucose. There are a lot of different solutes that make up a given bodily fluid. So it's going to end up being kind of a headache for us if we have to keep track of all of those individual concentrations. So what ends up happening for us is it is a lot more convenient for us to just have one metric that covers all of those solutes simultaneously. And that is what we mean when we talk about osmolarity. So to establish what exactly we mean by osmolarity, let's look at a specific example. So it says, imagine that we dissolve 2 moles of solute A. Don't worry about what the solutes are, just let's call them A, B, C, and so on. We take some water and we dissolve 2 moles of solute A, 1 mole of solute B, and 4 moles of solute C in the same liter of pure water. So we've got a beaker that has a liter of water in it, and we take these 3 different solutes, we put 2 moles of A, 2 moles of B, 4 moles of C, and we mix them all up in the same liter of water. Now, if we're keeping track of the individual concentrations of each solute, the concentration for A would be 2 moles per liter. We have 2 moles of solute A dissolved in 1 liter of water. That is a 2 molar solution. Similarly, for solute B, the concentration would be 1 mole per liter. 1 mole of solute B per 1 liter of water. And then for C, 4 moles of solute C per 1 liter of water, we end up with a 4 molar solution. Now, keep in mind, molarity, which is a concentration measure, only keeps track of one solute at a time. So for the same solution, we've got these 3 different concentrations. But for determining whether osmosis is going to occur or not, we need to keep track of all the solute concentrations put together, because that is how we determine what the water concentration is. So osmolarity is actually a very simple calculation. All you do is you add up the individual molar concentrations of each individual solute in solution, and whatever you get at the end is going to be the osmolarity of that particular solution. In other words, the total solute concentration in solution. So we would take the concentration of A, which is 2, the concentration of B, which is 1, and the concentration of C, which is 4, add them together, and we get an osmolarity of 7 osmolar. That's how you pronounce it, osmolar. So this would be a 7 osmolar solution, and that's not necessarily going to mean anything on its own. We have to infer from that by comparing it to the osmolarity of a solution on the other side of the membrane, and whichever one has the higher water concentration or the lower water concentration, that is going to determine in which direction water is moving. Don't forget, water moves from high water concentration to low water concentration. So if you have your osmolarity figure, you can infer that the higher the osmolarity gets, the more solute, the more stuff that is in solution, the lower the water concentration. And we can also wonder that if we put even more solutes in solution, if we add solute D and solute E and solute F into the mix, the osmolarity is only going to go up and up and up, and the relative water concentration will go down and down and down and down. Okay, so there is one caveat that we need to provide here. You're not necessarily going to include every solute concentration in your osmolarity calculation. Specifically, you are only going to consider what we call non-penetrating solutes. By that we mean solutes that cannot cross the plasma membrane. This is what we meant when we said earlier that osmosis only applies when you're dealing with a solute that cannot cross the membrane but water can. So for a given solution, we are only going to consider solutes that by themselves cannot cross the membrane, cannot participate in simple diffusion. So this is going to include things like sugars and salts and proteins. In a previous video, we talked about the rules that dictate what gets through the membrane all by itself and what does not. So this is only going to apply to what cannot. So penetrating solutes will include all those small nonpolar solutes that we talked about before that can cross the membrane all by themselves. So these are the types of solutes that if we do have a concentration gradient, they can move across the membrane all by themselves to correct the gradient so water does not have to. So in this case, we're talking about things like gases like oxygen and carbon dioxide, urea, and then hydrophobic lipid molecules like cholesterol and steroid hormones. So those are the types of solutes that, even if they are in solution, you are not going to include those in your osmolarity calculation. Okay, so let's talk about something. If you're dealing with osmolarity calculations, something special that you're going to have to do if you are dealing with a salt. So recall from Chapter 2 that a salt is a compound that is held together by ionic bonds. And we know from Chapter 2 that if you dissolve a salt into water, that salt is going to become dissociated. Water's ability to form hydrogen bonds will split that salt up into positively charged cations and negatively charged anions. Remember, we said that if you put table salt into solution, you're not going to actually have any intact sodium chloride anymore. It's all just going to be individual sodiums and individual chlorides. You can see this picture, which probably looks familiar from Chapter 2. So the thing to remember that after this dissociation occurs, osmolarity is all about individual solutes. So if you put a mole of sodium chloride into solution, you're going to then generate a mole of sodium ions and a mole of chloride ions, essentially meaning that whatever the original concentration of sodium chloride was, you're actually going to end up with double the osmolarity. So a 300 millimolar sodium chloride solution contains 300 millimolar sodium and 300 millimolar chloride because of the dissociation, meaning that if you add those two together, you've actually got a 600 milliosmolar solution. If we look at another example, say for magnesium chloride, if we start with a 100 millimolar magnesium chloride solution, after dissociation, we will have 100 millimolar magnesium, and because there are two chlorides there, we end up with 200 millimolar chloride for a total osmolarity of 300. Okay, so when you're dealing with bodily fluids, thankfully, most bodily fluids in the body have a predetermined osmolarity, and I'm not usually the type to ask you to memorize stuff, but this is one that I do want you to commit to memory. The osmolarity of most of your bodily fluids is 300 milliosmolar, or you could also say 0.3 osmolar. It means the exact same thing. So the reason why osmosis usually does not occur in most instances is because we do exist in a state of osmotic equilibrium between the extracellular fluid and the intracellular fluid. Now, we do know that there is a greater volume of water contained in the intracellular space, but the osmolarity is still going to be the same between the two compartments, because in this case, even though there is twice as much water in the intracellular compartment, there is also twice as much solute, so the water concentration still ends up being the same. 600 divided by 2 gives you 300 milliosmolar. 300 divided by 1 also gives you 300 milliosmolar. So because there is no difference in the osmolarity across this membrane, we exist in a state of osmotic equilibrium, and net osmosis is not going to occur. There will be a little bit of water transport back and forth, but no net osmosis, no net effect on the water concentration on either side. Now, a term that you probably are going to encounter occasionally is something that looks an awful lot like osmolarity. You might even wonder if it is a typo in your book. It actually is not. Osmolarity is not quite the same as osmolality. It is very similar, but it is just not quite the same. So what osmolality is the amount of solute, the number of moles of solute dissolved in a kilogram of water. That is not the same as osmolarity, which is the number of moles of solute dissolved per liter of solution. So even though the osmolarity of most bodily fluids is 300, you are probably going to see osmolality concentrations that are less than 300, something maybe around 275 to 290. For the sake of this class, we are going to use osmolarity, so please make sure that you are not accidentally using osmolality. It is important for a lot of your assignments you're going to be completed. Like I said, because the osmolarity of most of our bodily fluids are equal, in most cases there should not be a reason for net osmosis to occur. So osmosis will only occur if there is a difference in osmolarity between two different bodily fluids separated by a plasma membrane. The most common place that this is going to occur, you may have already figured out, is going to be the kidneys, because the kidneys function a lot in reabsorbing water from an initial filtrate to make sure that we don't pull too much water out of the blood. Now, when we're dealing with osmolarity, as we have mentioned several times, it is all about comparing the osmolarity of two different fluids, of two different solutions. So to help with these comparisons, we actually have three terms that we are going to learn about now to indicate the relative osmolarities of each solution. So look at this table for right now. We've got three different solutions that we are dealing with. Solution A contains 1 osmolar glucose, solution B contains 2 osmolar glucose, and solution C contains 1 osmolar sodium chloride. So these terms that we are going to introduce here are terms that you may or may not use when you are comparing two different solutions based on their osmolarities. So first, let's start by comparing solution A to solution B. You'll notice right away that both solution A and solution B utilize the same solute, which is glucose, but they have different concentrations and therefore different osmolarities. Because solution A has a lower osmolarity than solution B, we would say that solution A is hypoosmotic to solution B. Hypo means less, and osmotic obviously refers to the osmotic potential of that solution. So because solution A has a lower osmolarity than solution B, we would say A is hypoosmotic to B. We could also turn that around and say that B is hyperosmotic to A. Hyper meaning greater than, so that's just the opposite term that you would use if the solution is greater than the other one. Okay, so now let's bring solution C into the mix. Now, What I will caution you with this is that for solution C here, we are assuming that the dissociation has already occurred. So there's actually no need to double the concentration here as I showed you on a previous slide. So we can just assume that the osmolarity here is already at 1, and that's the highest it's going to be. So if we're comparing solution A to solution C, they have different solutes, but they have the same concentration, the same osmolarity. So we would say A is isoosmotic to C, and C is isoosmotic to A. So even though these are different solutes, if we put solution A on one side of a membrane and solution C on the other side, there should be no net osmosis occurring because the osmolarities are equal. Okay, so when you're calculating osmolarity, we are considering the content of a particular solution. We need to consider not only how many solute particles there are, meaning how many moles of solute are there, but also what is the total volume of solution. So when you're calculating osmolarity, that's usually going to be the best way for determining whether osmosis is going to occur or not. If you have a difference in osmolarity, that's going to be most of what you need to know. But the outcome that we really care about is actually what is called tonicity. Tonicity describes the effect a solution has on a cell if that cell were submerged in the solution of interest. So in most cases, this means you can assume that the ICF of that cell is going to be 300, and the extracellular fluid osmolarity is going to be that of whatever solution you are interested in. So the most common application of this for you all moving forward, assuming you're going to start working in a healthcare setting, is IV solutions. So IV bags contain various types of solutions that have different compositions in terms of solute, and therefore they're going to have different osmolarities. And because these IV fluids are ported into a patient's bloodstream, they actually have the potential to change the osmolarity of your patient's blood depending on what kind of IV solution you are providing. So being able to know about osmolarity and being informed about the potential risk or change to your patient is actually very important to understand. Especially if you're porting into the blood, we need to be able to understand the potential effects on red blood cells, since those are particularly important since they function in oxygen transport, and we've talked before about how important oxygen is. Okay, so we know that if the solute concentration of a bodily fluid gets too low, meaning less than 300 milliosmolar, that is going to generate a concentration gradient for water. So if we assume the ICF is at 300 and the extracellular fluid is less than 300, we have an osmotic gradient. And since water will move from high water to low water, or low osmolarity to high osmolarity, this means water is going to move from the area of low osmolarity, which is the extracellular fluid, and it's going to enter the cell by osmosis. So what you can kind of imagine is as a cell takes on water, the cell is going to start swelling up because the volume of the intracellular compartment is getting greater and greater and greater. And this can potentially cause the cell to, if the osmotic pressure builds up enough, to burst, which is called lysis, and that will kill the cell and cause all of the contents of the cell to leak out, and that is that. So solutions like this that cause a cell to swell up and potentially burst are called hypotonic solutions. And hypotonic solutions are usually also going to be hypoosmotic solutions when the comparison is between the hypoosmotic solution and the intracellular fluid. The opposite scenario would be if the extracellular fluid has an osmolarity that is too high, meaning much greater than 300, water will instead leave the cell, and that causes the volume of the intracellular compartment to go down. The cell will appear to shrivel up, and that is called crenation. So lysis for hypotonic solutions and crenation for hypertonic solutions. Both are bad and both are very detrimental to the ability of red blood cells to do what they're supposed to do. The situation that we desire, though, is to have an isotonic solution, meaning the osmolarity of the solution of interest is exactly the same as the osmolarity of the red blood cell's intracellular fluid. If that's the case, we establish osmotic equilibrium, and there is no reason for water to exit or enter the cell in wholesale fashion, and the volume and shape and integrity of the red blood cell should be maintained. Okay, so here's a table that actually lists a couple of very common IV solutions that you'll probably encounter throughout your career. By far, probably the most common one, one of the most common ones anyway, is 0.9% saline. This is also called normal saline. You can do some of the chemistry calculations here to convert from percent solution to molarity, but essentially what you're going to find here is that the osmolarity of a solution of normal saline is 300 milliosmolar. So for that reason, the osmolarity of 0.9% saline ends up being isotonic to the ICFs of red blood cells, or isoosmotic, I should say, and it therefore ends up also being isotonic. So normal saline is a good thing to give patients if you are just trying to keep them well replenished with fluids and you don't want to change the osmolarity of their blood. Giving a patient an IV bag that is just pure water is a big no-no, because the more water you add, the lower the osmolarity of their blood plasma is going to get, and that is going to cause lysis of red blood cells. So if you have, say, a patient that is not necessarily dehydrated, but you want to keep them hydrated, you want to give them the normal saline. You can't just pour pure water into their bloodstream. The next one on the list is actually normal saline, but it actually has an additional solute in it. We have 5% dextrose, which is actually just the same thing as glucose. Okay, so we already know that 0.9% saline is isoosmotic. So if we add another solute on top of that, we actually are going to end up with a hyperosmotic solution. So 5% dextrose is also 300 millimolar, so if you add the two together, the osmolarity of D5 normal saline is actually 600 milliosmolar. And since that is greater than 300, we would consider this to be hyperosmotic to the ICFs of red blood cells. But look over here, the tonicity is still isotonic, meaning that if you give a patient D5 normal saline, we are not going to cause any changes in the shape or volume of red blood cells, meaning there is going to be no net osmosis. So what's that all about? How can we explain that? Well, the explanation actually comes from what happens to glucose in the bloodstream. Glucose does not stay in the bloodstream for very long. What's going to happen is your patient is going to start secreting a little bit of insulin, and that insulin will act as a signal telling cells in the body to take up that glucose from the blood, and that glucose will move into the intracellular fluid of those cells, meaning that that dextrose is not going to be a part of the bloodstream for very long, so we would actually mostly ignore it in the osmolarity calculation. So really, for a D5 normal saline solution, we can treat it as if it's just a normal saline solution. The dextrose would not count because it does not stay in the bloodstream for very long. 5% dextrose in water will be isoosmotic because, again, it is 300 milliosmolar, but it ends up being hypotonic for exactly the same reason. That glucose does not stay in the bloodstream for very long, so after the glucose leaves, you're basically left with just water that has been ported into the patient's bloodstream. So that's why I cautioned you against ever giving a patient an IV of just pure water, because that is hypotonic and it's going to cause red blood cells to swell up. Half normal saline, which is half the concentration of normal saline, this would be 150 millimolar sodium chloride, 150 milliosmolar. It's going to be both hypoosmotic and hypotonic. and then 5% dextrose and half normal saline will be hyperosmotic. It'll have an osmolarity of 450 milliosmolar, but still it is going to have a tonicity that is hypotonic. So in most cases, you can usually draw the same conclusion from both the osmolarity and the tonicity. If your IV solution is isoosmotic, it's going to also be isotonic. The one thing you have to watch out for is the dextrose. So the dextrose is really the only thing that can cause there to be a discrepancy between the osmolarity and the tonicity for the reasons that we just described. All right, that is going to do it for this lecture on osmosis and tonicity. Thank you for your attention, and join us next time. Our next video is going to focus on active transport. Thanks for your attention, and I will see you next time.